Hello, I am Dr. Aizaz back with another visual lecture. This time we are discussing the replicative cycle or life cycle of hepatitis B virus, right? So this is the liver and liver cell or hepatocyte. A single hepatocyte is shown in this diagram. Hepatocytes have numerous important functions, and one of the function is to secrete bile salts. These bile salts passing through the biliary canaliculi ultimately reach the bile duct and then secreted into, what is this? Intestine. Here in the intestine, it is secreted into second part of duodenum. Here in the intestine, it has to perform a important function in emulsification of fat, digestion of fat, right? Now 97% of this bile acids and bile salts, they are reabsorbed back, right? They are synthesized by hepatocyte from cholesterol, and 97% of them is reabsorbed. Liver has so many other things to do. It cannot always create these bile salts, right? So it will reabsorb and then resecrete them back. So through portal vein, it will come into sinusoid, liver sinusoid. Sinusoids are the capillaries of liver that have, and these capillaries have wide gaps in between them, right? And through these capillaries, it is then it re-enters into hepatocyte. And to re-enter into hepatocyte, it needs a special polypeptide, a special protein or special channel, right? And through this channel it enters into hepatocyte. Now this channel is a co-transporter. It takes this bile salt along with two sodium molecules. So here is the bile salt, then there is two sodium molecules. You know, sodium is more outside than inside the cell. So when sodium comes inside, this bile salt also comes inside the cell. And here then this bile salt is then again secreted back, and this cycle continues, right? What is the name of this channel? It transports sodium and bile salt, and it co-transports these. So this is sodium bile salt co-transporting polypeptide. Previously it was called sodium taurocholate co-transporting polypeptide. Sodium taurocholate co-transporting polypeptide, or NTCP, right? Actually this name is a misnomer. It is not sodium taurocholate co-transporting, it transport other substances as well, especially other bile acids and bile salts. But maybe because this was the first, taurocholate is a bile salt, right? Because it was the first bile salt that was observed that it enters into this through this channel, so it was named as NTCP. Why I am stressing on this NTCP? The reason being that this NTCP is the transport channel. It is the channel, it is a protein with which this hepatitis B virus attaches and gains entry into the cell. By the way, this bile salt, when it enters into enterocytes, in this intestine, and then enters back into hepatocyte, this circulation is called enterohepatic circulation. Entero mean intestine, hepatic mean hepatocyte, liver, enterohepatic circulation. And NTCP, right? Now let's see that how it gains entry, how this virus it gains entry into the cell, and what happens next with this virus, right? How it replicates inside the cell. Let's zoom this. So here is a single hepatocytes, a schematic diagram that I have enlarged, and along with that there is NTCP. So along with that, you know, most of the cell, they also have heparan sulfate proteoglycan. It has numerous functions that I'm not gonna go into detail here, right? So let's suppose this wandering hepatitis B virus, it comes into contact. It first attaches with this heparan sulfate, and then it first loosely attaches with heparan sulfate, and then it undergoes high affinity binding, tight binding with anti-CP, right? These hepatitis V virus, it has a special predilection towards liver. That's why it is classified in the family of Hepadina-viridae. Hepadina means it means hepatocyte, right? So after attaching with anti-CP, then it can gain entry into the cell. So first a gentle smooth touch, loose touch with the hairy area, and then penetration inside, right? Yeah, inside the hepatocyte, don't think of other things, right? Then there is a drug here, I will discuss this experimental drug called Miclodex. It's an experimental drug, Miclodex B. It resembles this pre-S1 molecule of hepatitis B virus, and it attaches here with this receptor with anti-CP receptor, right? And it blocks the entry of hepatitis B virus. As of now, this drug is undergoing clinical trials, right? And it seems a promising treatment strategy for entry inhibition of uninfected hepatocytes, right? So let's see what happens next with this, right? As it attaches with anti-CP, this poor unsuspecting hepatocyte, unaware of this unseen enemy, it thinks that something useful and nutritious is coming, and it tries to engulf that nutritious thing, that it thinks that it is nutritious, and it engulfs this nutritious thing, right? It pinches this part of membrane with which this virus is attached, right? And in this way, this virus comes inside the cell. This process is called endocytosis, and this endocytotic vesicle is now formed, right? Now inside this endocytotic vesicle, now virus is present, and now this virus, its envelope of this virus, it will fuse with the membrane of this endocytotic vesicle, and nucleocapsid core will be released. Nucleocapsid core will be released into the plasma, and this endocytotic vesicle, it will then again plastered back onto membrane, so that these receptors will be back there at their position, and they will perform their respective tasks, right? So I am right now just removing this endocytotic vesicle from this diagram, right? So I have changed the color of DNA because it was not visible in the diagram. So now that we have this nucleocapsid core means that there is capsid along with the nucleus. Nucleus means DNA and polymerase enzyme. Now you know, inside the cell there is something called nucleus, and nucleus has nucleus pores. Actually, these are not physical pores or holes inside the nucleus. There are special channels here, or special proteins here, which checks the entry of substances. It is a selectively permeable. It allow only certain specific substances to pass through it, right? Now this nucleocapsid core, it comes close to this nuclear pore, one of the nuclear pore, and here the capsid disintegrates, right? As the capsid disintegrates, this DNA along with DNA polymerase gains entry into nucleus of the cell. Now it is inside the nucleus. Now when it comes inside the nucleus owns this DNA. Nucleus thinks that this viral DNA is its own DNA, and it takes care of this DNA. How it takes care of this DNA? It thinks that maybe this DNA is damaged because there are gaps inside this DNA. So it thinks that maybe this DNA is damaged, and it thinks that because it is damaged, it tries to repair this DNA. It tries to repair this DNA. So through host DNA repair mechanisms, this partially double-stranded DNA is converted into completely double-stranded DNA. These gaps are filled, right? Mechanism is quite complex and beyond the scope of this lecture, right? And before that, this DNA polymerase must be separated from it, right? It must be separated because it is tightly attached with one of the strand. So before this both strand can be covalently closed, it must be separated from them, right? So when it is separated, and human repair mechanism, they convert it into covalently closed DNA. But here one thing I would like to mention, that this partially double-stranded DNA, it is of course circular DNA, and these two strands, these are not tightly woven around each other. These strands are not tightly woven around each other, these are loosely present. That's why it is called relaxed circular DNA or RcDNA, right? So through DNA repair mechanism, this RcDNA is then converted into covalently closed circular DNA. It is now covalently closed circular DNA or cccDNA, right? But what happens to viral DNA polymerase? We don't know for sure. Most likely it is somehow degraded by some proteases present here inside the cell, right? Maybe it is exported out of nucleus and then degraded by proteases enzyme, right? So cccDNA, it is formed by human repair mechanism. From cccDNA, of course, there are genes on this DNA, and these genes are then transcribed. These genes are transcribed into their respective mRNA molecules. And again here, this virus is on the mercy of human RNA polymerase, right? So human RNA polymerase 2 enzyme, it forms mRNAs, right? mRNA transcripts for HBX protein, for pre-S2 and pre-S1, pre-core and pgRNA. All these I will tell you later what I mean by this. But here, of course, now then they are exported out of the nucleus, right? They come out of the nucleus into cytoplasm. This pgRNA, what is this pgRNA? It is pre-genomic RNA. Actually, this one of the strand, that is template strand of this cccDNA, a complete template strand of this DNA is, this complete strand is transcribed into a long mRNA molecule, and that is complementary to whole of this strand. And this complete RNA is called pre-genomic RNA. It is called pre-genomic because it is precursor of genome of hepatitis B virus, right? From this pre-genomic RNA, of course, it has all the genes of viral proteins, and of course it is also having somewhere here, it may have the gene for viral polymerase enzyme. And when this gene is translated, so viral polymerase is synthesized. This viral polymerase quickly finds this pgRNA and attaches with this pre-genomic RNA. Why it attaches with this pre-genomic RNA? Because it has 8 to 10 nucleotide sequence that is complementary to this pre-genomic RNA, and because of this it quickly attaches with it, right? Also this pgRNA, of course, it has the gene for capsid protein. So capsid protein is also synthesized. not just one capsid protein but a number of capsid proteins. Several capsid proteins are synthesized, and these capsid proteins, they will find this pgRNA polymerase complex, and it will associate with this pgRNA polymerase complex, and it will form a special icosahedral geometry around it, right? So this is icosahedral capsid core. Inside it is the pgRNA with polymerase. Now here what this polymerase will do, this polymerase with complementary to this RNA, it will synthesize a DNA strand. So this was RNA strand, complementary to this RNA, it will synthesize, it has now synthesized DNA strand. After this, it will destroy this, and this process is called reverse transcription, right? After this DNA strand is created, it will destroy the RNA through its RNase activity, right? So it has three activities. One is reverse transcriptase activity, second is RNA dependent DNA polymerase activity, right? And third is RNase activities. Interestingly, this is an amazing enzyme. It does not even need a primer, right? Because it has its own 8 to 10 nucleotide sequence which acts as a primer, right? So it has created single DNA strand. It then start creating second strand. But before it creates the second strand, it hastens to come out of the cell, right? So the second strand, it remains incomplete. There are gaps inside this. And when it comes out of the cell, it cannot get access to raw material for DNA synthesis. By raw material, I mean nucleotides. So it cannot complete the second strand. Second strand will again be synthesized by human DNA repair system, right? Now it must come out of the cell, right? But before it can come out of the cell, it needs something else. What is that? Yeah, it needs to get its envelope, right? So now we will see that how it gets its envelope and how it comes out of the cell, right? What is this? This is endoplasmic reticulum, right? Endoplasmic reticulum has ribosomes. These surface proteins, their mRNA, they attaches with these ribosomes, and these ribosomes, what they do, they translate these mRNAs, right? And these mRNA, these translated polypeptide are then processed inside the lumen of ribosome, and then they are embedded on their membrane because they are hydrophobic. So surface proteins are of hepatitis B virus, they are processed inside the lumen and then embedded on the membrane of endoplasmic reticulum, right? Now what will happen? Now here we have, but before that, let me tell you something else. What is this? This is pre-core mRNA. What do I mean by pre-core? Actually, core means capsid, right? Somewhat here is the core gene. What is this? Core gene. And before core gene, there is a small sequence of nucleotide which is called pre-core region. When translation starts from this part and ends in here, it starts from the start of pre-core region and terminates at the end of core region. Whole this polypeptide that is formed, whole this protein that is formed, it is E protein or E antigen, right? So pre-core mRNA leads to synthesis of which protein? E antigen, right? So pre-core, it again associates with ribosome of rough endoplasmic reticulum, but this time after processing the protein, this protein is small soluble protein. It is not embedded on surface, on membrane. This protein is dissolved inside the matrix. So here we have got E antigen inside the matrix, right? Now this nucleocapsid core, it has special signaling mechanism through which it finds the area of endoplasmic reticulum that has these embedded proteins. It finds the area of endoplasmic reticulum which has these embedded proteins, and it will enter inside the endoplasmic reticulum in this area, at this area, right? And then it will pinch outward and this will form a virus particle. Now this virus particle has to come outside. It has to come out of the cell. Here we have, what is this? Golgi apparatus. Golgi apparatus, this virus tricks this Golgi apparatus into thinking that this is body's own protein. This is a special protein or special hormone. This virus is special hormone or special substance that is synthesized by hepatocyte. So it packs into special vesicle or gift package, Golgi vesicle, and it exports it outside the cell by the process of exocytosis. It gains entry by endocytosis. It gets out of the cell through exocytosis, right? So here it got out of the cell, right? Now let me tell you something interesting. Something interesting about this virus is that even if the nucleocapsid core does not come here, this embedded surface protein, this empty surface protein can come out through the Golgi apparatus, right? And these empty non-infectious subviral particles are formed. Circular subviral particles are formed. Also there are subviral empty longitudinal particles are also there that I have not shown in this diagram, right? And E proteins, E antigen, they are also exported outside the cell, and because they are small soluble proteins, they gets dissolved into plasma, into blood of the infected person. Interestingly, these subviral particles and E antigen, they are produced in much greater quantity as compared to these complete infectious virus particle or DANE particle. Exact reason is not known. Maybe the synthesis of DNA takes time, or maybe nucleocapsid core, they are synthesized less as compared to other proteins. Maybe there are special transcription factors for these other proteins inside the liver, right? One of the reason for predilection of this hepatitis B virus towards the liver is, of course, one reason is because it has anti-CP receptor. The other reason is that liver has special transcription factors, right? Transcription factors for this virus, right? Maybe because of these transcription factors, these other proteins are quickly transcribed, right? So this was the replicative cycle of hepatitis B virus. But here I will tell you another thing about HBx. From HBx mRNA, HBx protein is synthesized. HBx protein is an oncogenic protein. It can, especially in chronic hepatitis B virus, it can trigger pathogenesis of hepatocellular carcinoma. Also it acts as transcription factor for viral proteins, right? So let's review the life cycle of or replicative cycle of hepatitis B virus quickly. So this was the hepatitis B virus. First, loose attachment with low affinity binding with heparan sulfate, then high affinity binding with anti-CP, right? Then it gains entry into the cell by endocytosis. It then fuses envelope of this virus. It fuses with endocytotic membrane, endocytotic vesicles membrane, and nucleocapsid core gains entry into cytoplasm. Here at the nuclear pore, capsid disintegrates, rcDNA gains entry into the nucleus. Here this polymerase separates, and rcDNA through human DNA repair mechanism is converted into covalently closed circular DNA, cccDNA, right? cccDNA, from cccDNA different RNA transcripts are formed, and one of them is pgRNA or pregenomic RNA. This pregenomic RNA, it is the precursor of genomic DNA, and this polymerase is also synthesized from it, and capsid is also synthesized from it, right? Through reverse transcriptase activity of polymerase of this viral polymerase, right? This pgRNA is first converted into single-stranded DNA and then partially double-stranded DNA. Nucleocapsid core then gains entry. No, first what happens? First, these surface proteins, these are synthesized by these ribosomes of endoplasmic reticulum, right? And embedded into this. E antigen is also synthesized and dissolved into matrix. This virus, it then gains entry, nucleocapsid core gains entry into this part of endoplasmic reticulum. Here it pinches outward, and then it goes to Golgi apparatus, and it is exported out through exocytosis. Spherical and longitudinal subviral non-infectious particles are also excreted out, and E antigen is also synthesized and exported out of the cell. By the way, these pregenomic RNA and most of the other proteins, these are translated from these mRNA, these are translated by ribosomes that are present inside the cytoplasm, right? Ribosomes, they can be present inside the cytoplasm. These are called free ribosomes, or ribosomes, they can be attached to endoplasmic reticulum, and these are attached ribosomes. And endoplasmic reticulum, which have these ribosomes attached to it, it is called rough endoplasmic reticulum, right? So this was about the replicative cycle of hepatitis B virus. I know it is complicated, but I have tried my best to simplify it, right? Of course, HBx, you know about HBx, right? It is a oncogenic protein, and it has role in, it acts as transcription factor. Finally, a few words about modes of transmission of this virus. It is a blood-borne virus, right? It means that for a person to get infected with this virus somehow must be inoculated into his blood, right? How it can happen? Blood transfusion. Let's suppose this was the commonest route of transmission of hepatitis B virus in past. Let's suppose if we transfuse the blood of a hepatitis B infected person to a normal person, this virus will gain entry into that person's blood, and he may be infected with this virus, right? It was commonest route, but right now these incidents of these accidents have quite reduced drastically because of the standard procedure of screening of hepatitis B virus before blood transfusion procedure, right? This virus, it is quite stroudy and it is quite sturdy and strong as compared to other enveloped virus. I don't know the exact reason, but it is strong and it can survive for up to 7 days on physical object. Let's suppose this is a hypodermic needle, right? And this hypodermic needle is contaminated with this virus particle, right? It can survive on this up to 7 days, right? And unfortunately, if we inject this needle into a person, he may get infected. That's why it is quite common in IV drug abusers, right? Because they share the needles, right? So also it can be inoculated through surgical equipment and dental equipment, right? IV drug abuse and personal hygiene equipment. For example, if a person goes to barber and sterilization mechanism is not adequate, through these razors and all these things, because most of the time micro abrasions are there on the skin when these razors are used, so it can transmit the hepatitis B virus through body fluids. Now let me tell you something interesting, that let's suppose this is a gland, right? And in this gland the glandular secretions are secreted, right? And raw material is taken from the blood. So from blood, secretions are secreted into this gland, right? Now this virus, because it is a small particle, it can also extravasate outside into this, into this glandular secretion. So in body fluids of the patient, especially who is actively replicating this virus inside the body, in his or her body fluid, this virus is usually present, especially in vaginal fluid and in seminal fluid, right? So because of this reason, due to sexual contact, this virus may be transmitted, especially through anal sex, because in anal sex they have observed that there is more chances of micro abrasions on the anal mucosa. While anal sex, there is more chances of formation of micro abrasions on the anal mucosa, and on these micro abrasions this virus may be lodged. This virus may be lodged on these micro abrasions and it may gain entry into blood, and in this way it can infect the cell, right? Now my question is that can it gain entry through the intact mucosa? About intact skin is it provides sufficient barrier to prevent the entry, right? So no question about the skin, right? But about intact mucosa, it is controversial. We don't know for sure that can it gain entry through intact mucosa or not. But what they have observed is that even through deep kissing with the patient, the virus is not transmitted, right? Maybe because in saliva this virus is less, or maybe because intact mucosa prevents the entry of virus. Exact reason is, again, I cannot say for sure that it prevents the entry or not, right? But theoretically, as it extravasate from the epithelium of this gland, theoretically it can intravasate as well, right? But I don't have any research articles or data to back up this opinion, right? If you have some idea about this, I will love to hear from you. Thank you so much for watching this lecture. Please give your precious comments about this lecture and please subscribe to my channel. Also, please check out my Facebook group. When this lecture series on hepatitis B virus will be completed, I will share the lecture notes of these visual lecture series on this group, right? Thank you so much.